Okay, let's get started. I'm very glad to have today Ajay Chitnis and Abhishek Mukherjee from the NIH, who are going to be talking about their work on modeling the formation of rosettes in zebrafish lateral line. And I put in the chat a link to the code which you can download to run their examples. And I'm not going to take any more time from them. I'm going to turn over the screen share to them.
Thanks, James and Hayden, for giving us the opportunity to share some of our work with you today. I'm going to actually give you quite a lot of biology background for the motivation for our studies, what the kind of modeling we actually did before we got to CompuCell and why we needed to do that. And as you will see, actually, this morning's session was probably, I couldn't have had a more perfect introduction to what we did, because the model that we developed is really based on pretty much the exercise that I guess Abhishek did in a previous session. So anyway, I'll get started. So the big question, big question that our lab is really interested in is trying to understand how interactions between cells determine the specification of fate, cellular organization, and movement to determine the self-organization of organ systems in a developing embryo. Now, this is a big question, so we tackle it by studying a relatively simple and wonderful system called the zebrafish posterior lateral line primordium. So the zebrafish lateral line primordium is this group of about 150 cells that migrates from near the ear to the tip of the tail in the zebrafish. And as it migrates, it deposits these sensory organs called neuromass. And by the end of about 48 hours, it has reached its destination and deposited these seven neuromass. And later in development, the number of these neuromass will continue to grow. So these neuromass are, as I said, sensory organs that have these sensory hair cells that stick out from the skin into the water. And these sensory organs help the fish detect the pattern of water flow over their body surface. As it migrates, what this slide shows is that the primordium is migrating over the surface of a muscle layer, the somites, along this groove called the horizontal myoseptum, and it's migrating under the skin, so that during this migration, it is confined under the skin and the somites. And as you see here, as it's migrating, it is forming these epithelial rosettes, and you see one of these deposited neuromass. Now, if we look at this at higher resolution, you can see that there are many more interesting features that we had actually not recognized before we were able to do this kind of microscopy. You can, in the sagittal section, see the structures where these tall epithelialized cells have made these apical constrictions to form what we call epithelial rosettes. And in addition, these kinds of studies allowed us to recognize a new class of cells that sits on top of the deeper cells that are forming these rosettes. And what's special about these cells is that both the superficial cells as well as the basal feet of these tall epithelialized cells extend lamellipodia, which allow this slug-like creature to migrate under confinement between the skin and the extracellular matrix below. And so together, these studies have allowed us to define all the different types of distinct cells with distinct shapes and behaviors that are maintained through the course of migration. Now, I should say that I know that you are all from different backgrounds, so if there's something that's not clear, just please stop me and ask some questions, or put a message in the chat that James can let me know about. Okay, so now as this primordium migrates, there are two important things happening. Number one, all the cells are moving collectively together. At the same time, there is the periodic formation of these protoneuromass, or primitive neuromass starting from the trailing end of the primordium. And the formation of these protoneuromass involves two processes. One is the reorganization of the cells to form these clusters, and the specification of a cell at the center of the cluster that will express a transcription factor called ATO1A, which simply gives these cells the potential to eventually divide into two cells that will form the first two sensory hair cells of this sensory structure. Now, the formation and the coordination of many, many events in this developing system are coordinated by the polarized activity of two signaling systems in the primordium, Wnt and FGF. So Wnt and FGF are simply signaling factors that activate their receptors in neighboring cells to influence their fate and behavior. And one important difference that might be of consequence for many of the things that I'm going to tell you is that what distinguishes these two secreted factors is that Wnt can only travel a very short distance from each cell that is producing it, while FGF can travel a much further distance. So while cells in the leading domain produce the FGF, they can travel further to the trailing part. Oops, I forgot to say. So it's going to, these signaling systems are going to interact, and they are going to coordinate both the formation of these protoneuromass, which includes the reorganization of cells to form epithelial rosettes, as well as the specification of a central cell as an ATO1 expressing cell, and also the collective migration of the system. So let's start with the collective migration. It turns out that primordium migrates along this route following a stripe of the expression of a chemokine called CXCL12A. It acts as a chemoattractant for the system, but the manner in which it works is quite interesting and is regulated by this polarized Wnt and FGF activity. The stripe of CXCL12 that defines the route along which it migrates is relatively uniform, and the reason the primordium migrates in a particular direction is because Wnt and FGF activity regulate the expression of two different types of chemokine receptors. So at the back is one of these receptors whose job it is to degrade the chemokine which is in its path. This degradation creates a self-generated gradient, which now these leading cells are able to respond to and migrate. At the same time, remember I told you that the Wnt cells produce FGF? So it turns out that the FGF that is secreted by these leading cells towards the trailing cells also acts as a migratory cue. So they move towards the leading end by following this FGF cue. So this is how both Wnt and FGF collaborate to allow chemokine and FGF signaling dependent migration to determine this directed migration of the primordium. The interactions between FGF and Wnt also coordinate the formation of these rosettes and the central ATO1 expressing cell. So to understand how that works, we need to add a few more elements to the regulatory network that I have already begun to describe. So we already know that Wnt dominates in the front and it produces the FGF that is able to activate the FGF receptor in the trailing domain. At the same time, the Wnt activity initiates expression of a number of factors that inhibit the activation of the FGF receptor within the cells that are actually producing the FGF. And at the same time, these trailing cells in which the FGF activity has been initiated, they drive the expression of additional factors that inhibit the activation of the Wnt system in the trailing part. Now, it turns out that this relatively simple looking set of interactions has some remarkable properties. It constitutes together a reaction-diffusion system, which is a version of a class of patterning systems described by Alan Turing and Gerer and Meinhardt, where there is local activation coupled by long-range inhibition. Now, this is not as obvious in this network, but the reason this happens is that Wnt is doing two things. it is locally inhibiting the expression of its inhibitor, and this actually contributes to its local activation. At the same time, at a long range, it is activating the FGF system, which acts as an inhibitor. So there is local activation and long-range inhibition. And some years ago, I realized that simple set of interactions can initiate the spontaneous emergence of these different patterns, as illustrated with the simulation that we built many years ago using another agent-based modeling environment called NetLogo. And I'll actually be using NetLogo for a number of the illustrations for some of the phenomena before we get to CompuCell. So based on this set of simple simulations, we were able to see how these different patterns arise as a consequence of the simple set of interactions. And we recognized the similarity of this network to the ones I have just described. And this allowed us to understand how a set of interactions mediated by the system could initiate the formation of the center-biased spot of FGF activity at the trailing end of the primordium. This is not really going to be part of the CompuCell model that we're talking about, so I'm not going to go into much detail about it, but you can ask me more about it at the end if you'd like to learn more. Anyway, the idea is that we realized that this center-biased spot of FGF activity helps to initiate both the formation of these epithelial rosettes, and importantly, the center-biased pattern of activity helps to specify the central 801 expressing cell, because this center-biased activity initiates center-biased 801 expression, and that is in the context of lateral inhibition, where each of these 801 expressing cells tries to inhibit its neighbor from doing the same thing, results in the restriction of that 801 to a central cell. So based on our understanding, we built a model using NetLogo, where this represents an initial group of cells in the primordium, and there's basically a set of interactions based on this network that I showed you. And in this model, there's the cells are dividing, the system is growing, and as the Wnt system shrinks, this simple model illustrates how spots of FGF activity will spontaneously arise that in this particular version of the simulation allow you to see that when the FGF signaling represented in blue persists for long enough, the cells make spring-like links with their neighbors to simulate the formation of these epithelial rosettes. In another version of this simulation, we implemented the process where the center-biased FGF signaling initiates 801 expression, and we had a network representing lateral inhibition mediated by notch, and that allowed us to see how in the same context, a center-biased, a central 801 cell would be specified in this context. So all this was great, and we were very happy with the framework that we had developed for understanding how these interactions between FGF and Wnt help to coordinate the periodic formation of these epithelial rosettes with central 801 expressing cells. But as we played around with the system, we came across a rather interesting observation. And it's related to how the number and size of these rosettes change when you interfere with the mechanism that selectively determines the migration of either the leading cells or the trailing cells. Now, remember, I told you that the leading cells move in a particular direction in response to this self-generated chemokine gradient. Now, one of the things we can do is we can transiently exaggerate the expression of CXCL12A. So now that it's broadly expressed at very high levels. And when that happens, the directional information imparted by this system is lost, and the leading cells actually stall because they don't know what direction to go in. And so it's just done for a short time, and then eventually the system recovers. In the course of such an experiment where we have transiently stalled the leading cells or slowed them down, something very interesting happens, which is going to be shown in this video. So this is going to be a video of showing the membranes of the migrating Primordium. And this is a PIV analysis which shows the relative speed of the cells in the different domains um, um, uh, within the Primordium. And what you will see is at some point, the leading domain will start to slow down right now. And as it slows down, two of these constrictions representing the epithelial rosettes come together, fuse to form a large rosette. And then as the migration resumes again, it splits to form two rosettes once again. So these observations made us realize that while there is a very elaborate system that helps to generate this particular pre-pattern of FGF signaling to determine where the rosettes form, it turns out that in the end, the system is really all about mechanics. And if you interfere with the manner in which cells are pushing and pulling on each other, it can alter the number and size of these rosettes. So to explore this idea, we built another NetLogo-based model, in which case now all the Primordium cells are just represented by little balls that are connected to their neighbors by springs. And what you notice is something very interesting that happens in the system. So if you just allow these cells to interact with each other, it spontaneously forms these clusters, which when there's nothing to oppose it, they progressively fuse together to form larger structures. If you add migratory behavior to it, then it opposes the formation of these rosettes. And in fact, this phenomena is really a mechanical form of local activation and long-range inhibition, this patterning system that plays a recurrent role in many, many aspects of animal development. So the local activation happens in this model because each of these cells, which are called turtles in this programming environment, make spring-like connections with neighbors within a certain radius, and that pulls cells closer together, bringing more turtles within the range to make links, and that becomes a self-promoting process as the clusters form and then coalesce to form bigger and bigger clusters. However, you add to that migratory behavior, this creates tension, which is like long-range inhibition of this clustering, and the combination of these two processes leads to the spontaneous formation of these periodic clusters. So using this kind of framework, we built another model. Again, I'm not going to go into it in great detail, which allows you to see how you can start with a completely uniform block of cells. And we have different domains representing different degrees of spring strength to pull cells together in the green Wnt-like domain and the trailing FGF-like domain, and illustrates how this combination of local activation representing adhesion and constriction, the forces associated with this, which pull cells together, when it is coupled with tension associated with migration, allows the spontaneous formation of these clusters. So all this was great, but we were not able to take advantage of this sort of NetLogo model, which represents cells as simply points in space, doesn't allow us to capture the richness of the system where the cells are migrating between the skin and the muscle. We've already discussed the diversity of cells involved, including specifically these kind of superficial cells, which allow the system to migrate sandwiched like a slug between the skin and the underlying somites and extracellular matrix. And we wanted to build a more comprehensive model, taking that could integrate what we had learned about this complexity at multiple scales. And this is what brought us to the model that Abhishek is going to describe to you, where, as I said, what you learned in the morning really was the prelude to the model that we elaborated on to represent the primordium. So Abhishek will take you through the stepwise construction of this model. This is simply to illustrate the basic principles involved, where each of these different cell types that you have seen in this schematic are represented in this CompuCell model. There were three key things that allowed our model to recapitulate many of the features of the actual primordium. The first, as you learned earlier this morning, which was critical, is using a sort of stepwise logic to try to think about how each of the different types of cells adhere to each other. Second, we have these tall epithelialized cells in the FGF domain. These were represented, as we learned this morning, with three compartments, apical, lateral, and basal. And using an element that you weren't introduced to yet this morning, but I don't know whether you've done it earlier, but with these spring-like elements, we could incorporate apical constriction. And finally, we have the cells in the leading domain and in the trailing domain moving in response to these different migratory cues, the chemokines in the front and FGF in the back. And in the back, it turns out, as we said earlier, that once we have these FGF cells represented with these different compartments, it's just the basal feet of these tall epithelial cells that move in response to the FGF, and the superficial cells, which I've shown you here, which will contribute to migration. And all these things put together allowed Abhishek to make this beautiful model that he's going to describe. And I think that by the point at which we made this model, the notion that cells will spontaneously aggregate to form these clusters was not really the big surprise. We had already learned about this through earlier stuff. But I think that what really amazed me about this model was how simply the logical representation of differential adhesion between the different cell types, which Abhishek is going to describe to you, was adequate to account for this behavior of the model primordium, which we have been able to use to recapitulate many of the behaviors of the system. So that's really all I wanted to show you in this introduction, and I actually can stop sharing and take questions. This is actually another CompuCell model that I didn't describe today, but it just looks at the primordium from a different perspective.
It's a great example. I think the issue of how to translate a model that was implemented in NetLogo into CompuCell is an interesting one.
Yeah.
What the challenges are when you go from one modeling framework to another. It's not reflecting the biology, but ultimately, if you're reflecting the same biology, whatever the simulation package you use, you should get similar results.
Yeah, so I think that actually this is we built the CompuCell model to just visualize the kinds of things that I've just shown you, but really our plan is to go back to incorporate the reaction diffusion components into this model and develop a much more integrated model. So that's actually, I have a student coming who I'm hoping that he will do this. So there's a lot more we can do with this model. And it's sort of a platform that's not an endpoint, but it's sort of a place where, as we continue to do experiments, we keep adding to it, integrating parts to it. And I think as a biologist, the thing that I have learned that's been most valuable is when you build a model as a biologist, you get to confront what you actually know, what you don't know, what are the pieces that are missing. And throughout the trajectory of making these models, whether it's been in NetLogo or in CompuCell, I have had many, many instances where to get the system to work like the biological system, I had to add something in. And then somewhere down the road, we discovered the biology that corresponds to the thing that we had shoved in to our computational model.
Well, that's important. You'd like to be able to predict things as well as post-dict them.
Yes.
Okay, Abhishek, did you want to show us the code, and then we can have a more general discussion after that too? Do you want people to download the code and try running it?
Yeah, they can play with it and change the parameters and all. I have a couple of exercises in the end, which they can probably do if they are interested. Okay. Okay, so I'll start. So as Ajay already introduced, this is a side view of the model system that we work on, the posterior lateral line primordium, and it is migrating as per convention to the right. And as you can see in this image, there is a skin on top. And what you cannot see from this image is there is also an ECM, a thin layer of ECM at the base of the primordium, and below it there is and there are muscle cells or large muscle layer. So apical direction is towards the top, where you see these constrictions denoted as these high intensity spots and marked by arrows, and they are where several of these cells they converge together to form constriction. And overall, if you look at it from the top, then you see that these are rosette like structures. So this is a schematic which shows pretty much what we think about of the primordium, where there is less apico-basally polarized, or the constrictions are not formed in the leading domain, although they are still apico-basally polarized, but they just don't form constrictions as in the trailing end, which is governed by FGF signaling and its downstream factors. And then there are also the sheath cells, which have recently been discovered by our lab. And Ajay also gave you a demonstration on that. And yeah, and then there are the skin layer and ECM. So broadly, we divided the model primordium into two domains. In the leading domain, we have the blue or the non-apico-basally polarized domain, and in the trailing domain, we have the more apico-basally polarized domain. So we start out. This is an initial setup. If you run the model, you first see that this is the setup. We have modeled the skin layer on top, and we already assume that the primordium starts out as uh, a a system with two distinct divisions, and the leading domain and the trailing domain. However, in reality, that's not really the case. Where if you look at time-lapse movies, then the epithelialization starts gradually from the trailing domain and shifts more like a wave towards the leading domain. But here, for simplicity, we started out with a predefined epithelialized and non-epithelialized domains. And in between sandwiched are the sheath cells. So if you start out, this is just a screenshot of how the new CompuCell3D plugin looks like, a GUI looks like. So our domain is 800 pixels wide in X and 300 pixels wide in Y. And this is a 2D model, so Z is 1. And we wanted to make the primordium migrate freely and not get impeded by the X boundary, so we made it periodic and simulate a larger domain. And the Y boundary has no flux boundary conditions imposed on it. Yeah, and the initial cell layout is a rectangular slab-like layout. Average membrane fluctuations are 20, and pixel coupling, the neighbor order is 3. We did not change these units of space and time, because our purpose for this model was to simply explore the physics behind the constriction formation or the rosette formation, and how it opposes the addition, and how it opposes the migration. So step two, these are the various cell types that we wanted to incorporate in our model, apart from the medium. Here, like I said, FGF domain cells, they are pre-epithelialized, and they are composed of those composite cells, compartmental cells that Julia was talking about in the previous module. And no, we also followed, this model was inspired by Priam and Julia's model of the somite segmentation. And so we followed similar methods of how, when Julia was talking about uh, how to make each of these compartments uh, uh, part of the same cell, so that they don't get stuck to the neighboring cells, or they don't move around, but they behave as a single cell, different domains of a single cell. The reason we wanted to do that was this allows us the flexibility to assign different properties to the different domains. For example, we wanted the red domain or the apical domain cells to come closer to each other than the green domain cells, and we also wanted to assign migration properties specifically to the basal domain cells, because these are in our model, these are representing the basal feet. So these FGF epithelialized cells, they don't really migrate. I mean, if you want to model migration of those types of cells, they have basal feet, basal lamellipodia, which protrude from underneath, and that is how they migrate. So we wanted to model that. And the wing domain cells, we did not specify basal feet per se, but we simply assigned migration potential to these cells, which apply at the centroids. And then we had ECM layer underneath, and the muscle layer below. Please stop me and ask questions if you cannot follow, or you have some question. I'll be happy to answer. And then we wanted to assign various properties or constraints. And so these are the cell properties that we wanted to assign. The first and foremost is contact, and because we also have compartmental cells, we wanted to assign internal contact energy of these cells. And then we also, as I'll get to it, we also wanted to assign these spring-like links between cells, which allow contractility. And so that's the focal point plasticity links. I think that's what Priyam will talk about in the next lecture series next week. Yeah, and then we had volume and surface constraints. We did not want to modulate those or regulate those in the Python part of the code, and we just wanted to fix whatever was there, whatever we wanted in the XML part of the code, and that's why we just had the volume flex and surface flex plugin selected. And but we wanted to modulate external potential or the migratory potential of cells during runtime. So that's why, and that could only be done via Python, the Python part of the code. And so we had this external potential local flex plugin. And then connectivity plugin allows us to put a penalty on cells so that they don't break up. And they, while playing with this model, while making this model over many iterations, we found that certain cell types, like for example the apical cells, they when they contracted, they had tendency of breaking up, and so which imparted unintended consequences to the system. So that's why we had this global connectivity plugin. Yeah, so that's it for here. And then for we didn't have any diffusion, so we just skipped over this chemical fields window. And then when you finish making this model in the GUI, I won't show you the code here, but I want to take you through the assignment of various contact energy properties, which is really the crux of this entire simulation system. Um, so this is the schematic which I showed you earlier, and this is also the model setup. Here these are compartmental cells, FGF cells, with the apical, lateral, and basal domains. And here you see a cartoon of two cells side by side. And here you see the more unpolarized cells in the Wnt domain side by side. So what really went into thinking about how to assign contact energies to these various cell types is that we didn't want mixing between because in the primordium, essentially, if you look at it long enough, there isn't real interchange of cell neighbors, especially in a 2D domain. This is a 2D. Even in 3D, there is a bit of jiggling about of cells, but it's not very drastic, where cells, they exchange neighbors very frequently. So that's why this 2D model could be a good representation if we had cells relatively fixed in their positions with respect to each other. And so we wanted the sheath cells, which had recently been discovered, to be sandwiched between the primordium lower layer and the skin. And we didn't want that to really and we didn't also want the primordium to break off from the skin and into the, let's say, the medium. So to ensure that all of these things happen, like for example, now if we go just to the Wnt domain specification, contact energy specification with itself as well as with the other cell types, we see that we wanted the Wnt cells to remain relatively adherent to each other, more adherent to each other, and so we had low contact energy for that. Um, but we did not want um, uh, mm, uh, and also we wanted the interface, the Wnt on one side and the FGF lateral, the green domain on the other side, to not break up. So that's why we wanted the Wnt and FGF lat, which is the lateral domain, to have a very very low contact energy, so very high adhesivity, actually the lowest here among the Wnt versus the other cell types. So and we did not want the apical and the basal domains of the FGF system to be as adherent to the Wnt cells, so that they would be free to form rosettes at the top at the apical domains, and they would be free to migrate more in the basal domains. So yeah, and the sheath cells are and when this model was developed, sheath cells, and we still don't know a huge amount about sheath cells and their properties. We are still trying to figure that out. So this was kind of a relative gray zone, and so we wanted the properties of sheath cells to be like intermediate between the various Wnt and the FGF domain cells. So we have relatively higher contact energy between Wnt and sheath cells, so that the sheath cells are able to migrate about, which you also saw from one of the movies that Ajay showed. So sheath cells should be motile and they should move about, and so that's why we didn't want a high adhesion between the sheath cells and the other kinds of cells. Wnt and skin, we did not want the Wnt and skin cells to like really adhere to each other, otherwise that would impede migration, and so we have it relatively higher in terms of contact energy with respect to the Wnt and Wnt. And Wnt ECM was also relatively higher. And we did not want of course the Wnt and the muscle cells to interact ever, so that's why we set it quite high. Now coming to the apical domains, we wanted the apical domains of neighboring cells to be very adherent to each other, and so we set it to 1. Whereas we didn't want the apical domains to really contact the lateral and the basal domains, definitely not the basal domains, so that they don't get inverted. So we set the FGF API to FGF base contact energy really high. And but we wanted the apical domain to contact the sheath cells because they are in contact with the sheath cells. Even in the real system, we wanted that to be relatively lower, but not so low that they both stop migrating or they have really high adhesion between each other, among each other. So, and there are many parameters here. I'm just going to take you through a few of these. And we didn't want the apical domains to really come into contact with the skin or the ECM or the muscle. So we set those to very high. Lateral and lateral domains, we wanted them to be moderately adherent to each other, because we noticed that there was a kind of a room that was afforded to us by this model setup, where we could change it over a wide variety of ranges. And so we set it to a moderate value, and in a later exercise, we actually in a later simulation in the paper, if you look at that, we actually simulate one of the conditions where we had higher adhesion between the lateral domains, and you can actually look at that. And FGF lateral to basal domains, we didn't want them to be very sticky among the neighbors. Same with the sheath cells, and we definitely didn't want them to contact the skin or the ECM or the muscle. So those are really high. Basal domain to basal domain, we wanted them to be quite motile and not really want to adhere to each other. So we set that high enough, but not so high that they would sort of invert and migrate in other directions. So and then we have base to FGF basal to sheath cells, slightly lower, because these sheath cells, while they are migratory, they also when we set up this model, we also wanted to incorporate a deposition mechanism where the sheath cells also tend to get deposited alongside the epithelialized FGF cells, and so we didn't want that. I mean, that's why we set it slightly lower than the basal to basal, and basal to skin was set really high because we didn't want them to contact each other. The basal domains migrate on top of the ECM, and so that adhesion energy was set to be quite low. and yeah, muscle basal was also a bit high. And then sheath cells, to sheath cells, we wanted them to be motile and migrate. So it's moderately high, but sheath and skin, we wanted them to be in always in contact with each other. So that's a bit low. And sheath ECM is a bit high again, and we didn't want the sheath and the muscle cells to contact each other, and so on. And skin, we didn't want the skin muscles to skin cells to break apart, and so that's why we set it to very high adhesivity. Yeah, and skin ECM, they should separate from each other when the primordium migrates through. So it's set to a moderately high adhesion energy. ECM, we didn't again want them to break apart and have all sorts of unintended consequences in terms of the primordium breaking through the ECM, and so we had that to be very low. Yeah, so these were a lot of parameters, and I went through some of the rationale behind choosing the relative relative energies that we set, and how we went about going about those. So some parameters are I mean, quite in terms of uh, they have a high tolerance range in terms of the migration of the model. So you have the model, you can play with the various parameters and see what you think about these. And some parameters have higher effects than others. I won't go into all of those details. But and the other important aspect was the internal contact energy plugin, and we wanted in each of these compartmental cells, we wanted the apical to lateral domain energy to be really low, so that they are very adhesive, and lateral to basal to be very low, but apical to basal they should be quite high, so that they don't contact each other. And then we also incorporated the focal point plasticity links. Here you can see those in white. We incorporated links within each cell, apical to lateral and lateral to basal, and apical to apical in the neighboring cells, and lateral to lateral in the neighboring cells. We didn't have FEP links between the basal and basal domains. And we had FEP links between the Wnt cells also. So these are the relative parameters where the strength of links, or the springiness of the links, contractility of the links between the Wnt cells was relatively lower than, let's say, the Wnt and FGF lateral domain cells, because we wanted the lateral domain and Wnt cells to not sort of break off, which they had a tendency as a consequence of forming rosettes, local rosettes. And we had the target distance adjusted so that I mean, according to the cell sizes. And so these were the target distances, and we had a much lower target distance of the apical domain, so that they wanted to form rosettes. And we had lower max distances, so that these links also break up when they extend beyond a certain range. So that allows really the rosette formation. As I'll show you in the next few slides how it looks like. And what else? The skin, we didn't want the skin cells to break apart, so we also imposed these or implemented these FPP links within the skin, which had a very high max distance, and they also had high strength among them. And ECM, we did not need to implement such a high max distance or a high strength, because perhaps because the ECM had the muscle layer beneath, that this was not a problem of breaking up the ECM and migrating, the primordium migrating through, but it was rather major problem with the skin and the primordium trying to break through that layer. Yeah, and so the within the cell itself, within each of these compartmental cells, we had the apical to lateral and lateral to basal. We implemented these FPP links to also sort of simulate how or to maintain the structural integrity of these epithelialized cells, where there are many cytoskeletal elements such as microtubules which enable the epithelialization of cells and retain them in such a position. So we implemented these FPP links to sort of be proxies or to simulate that kind of an effect. And we set the target distances according to the domain sizes of these cells, the maximum distance also similarly. Yeah. Any questions until now? Okay. So upon implementing the FPP links, you see that what we intended was that the apical domains would constrict more than the lateral domains, and that these cells would also retain their uh, uh, integrity, structural integrity while migrating. So the other plugin that we used was the migration plugin. We implemented migration in the migratory forces or external potential in the basal domains of FGF cells and the Wnt domain and in the whole of the Wnt cell and the centroids. And we also incorporated this migration force in the sheath cells. So these were the parameters of the external potential or the forces that we used. We found that we had to impose a higher external force on the basal domains of the epithelialized cells, because they tended, if we had them similar to the Wnt domain Wnt domain cells, then they seem to be lagging behind, and the primordium would really stretch out, and eventually they would break apart. So, and this is something that we are also interested in exploring a bit more, because uh, physically seems to be in the real system, there seems to be reasons why the and previous work has also shown from Holger Knaut's group in New York, where they have shown that the epithelialized cells or the FGF domain cells, they tend to have more force, or they tend to apply more force on the underlying substrate, the ECM, while they migrate. So there seems, and although our model was kind of did not follow that particular rationale in that sense, but we also found that we also had to implement a higher migratory potential to the basal feet of these FGF cells than the Wnt cells. So yeah, that is an interesting physical aspect to explore a bit more. So here, if you go from top to bottom, you see a time course of the evolution of this model primordium, where we started out with this setup as I initially showed you. And then as time progresses, very quickly, you have the FPP links made between the domains that we implemented them in. And you also see that when these FPP links are made within the FGF domain within each of the cells and among the neighboring cells, you see that in the initial few time points, they undergo a lot of reorganization owing to the relative um, low value of their maximum distance over which, or the influence, maximum region of influence over which these links can operate. And so you see that it goes from like these two cells coming together and forming these FEP links, and then to three clusters of these cells. And if you look at this black rectangle here to and follow these, then you'll find that these also within them, they also undergo remodeling, constant remodeling as cells grow closer to each other and the apical domains come closer to each other and they are more contractile. They pull more cells into their neighborhood and their influence area. And so but over time you see that the relative number of these rosettes, and by rosettes I mean these cells which form these FPP links between them, between the apical domains, they become consistent over time. At a later stage, due to migration, they do change sometimes, but in these shorter time scales, they as the epithelialized domain that gets remodeled, they kind of retain their neighbors because they grow out of in the influence region of the others. And now if you follow the black rectangle again, so you see that there are two of these rosettes with three cells in one and two cells in one, and there is one single cell over here that gets remodeled into two and three there, and then two and four, and then these over time they constrict to form these nice rosette like structures. And while the primordium is also subsequently migrating. Uh, so this is an example video of control, and here I have as I play this video, you'll see magenta arrows marking the various rosettes over time. So you see four rosettes over time. And there was one example which Ajay showed in his during his talk, where we wanted to model how the primordium uh, behaves, or how the rosette remodeling takes place when the leading domain stalls because of perturbation of the chemokine gradient. And so this is an example of that, where you'll see that after a period of time the leading domain will get stalled. So here we impose a stalling of the leading domain, and that leads to fusion of these two rosettes into one. And once the primordium resumes migration, this rosette breaks up into two. This rosette will break up into two. Yeah, it breaks up there. I'll let it play again. So this modeling framework allowed us to incorporate a lot of these aspects that we wanted to model of the primordium, and also to model the relative interactions between cell addition and the constrictions as well as the migration. So Aritra is saying there is no movement of the cell layers. So, did anyone else try the model out?
When I run it as you've given it to us, it looks like you're in the stalled configuration.
Is it? Let me see.
So maybe there are some parameters we should try to change. If you suggest those, we can do that.
Yeah. Yeah, so basically if you go to the steppables file, let me see. The Python file. Can I share my screen?
We can certainly share Twitter if you want,
Yeah.
or you can share your desktop and show everything together.
Okay, let me see. You cannot see my
We
tweeted
can see.
screen right now,
We
right?
see Twitter.
Okay,
Yeah.
you see Twitter.
We
Okay,
see
cool.
Twitter.
Cool. So here, if you go to the steppables screen, this one. So basically, what we wanted to do was we wanted to also impose. We wanted to impose this. Does it have the cell lambda vec x parameter on there? Should be
Yeah,
-2.
we have -2. Yes.
Okay. Okay, let me try and play it.
Had you made a different version for doing the simulations that you saved by mistake or something?
Let's see. I'm just trying to play this one. So you see my simulation, right?
Yeah.
It is a bit slow, but it does migrate.
Well, so maybe we didn't wait long enough.
Yeah.
It's a much longer time than.
So that's why I mean in the videos it's approximately 20,000 MCS over which the Primordium migrates. So it takes a while.
So maybe we should add a plot of the position of the Primordium versus the time, and then we'd
Right.
see it. So I'm at my code is at 6,000 Monte Carlo steps, and the blue over the 400 X position.
Yeah.
So it's the main thing is we have to be patient.
Right.
Now, were there other
people
changes?
can play. Yeah,
Go ahead.
people can play with the external potential value, and so they can increase the value and see what effects it has. In some cases, if it's too high, then the domain might break off. That's why.
Would you be willing to show us one example of that? Maybe
Yeah.
show us how to make that change and then
Sure.
run it.
Sure. Let me see. So here, let's say in this step, I make it, let's say 20 and see if it really makes a huge difference or not. So it does seem to be migrating faster, the blue domain. It's quickly reached 400. And you see it's also really stretching out, and you saw that the FPP link broke between the Wnt and the trailing domain. So the trailing domain is simply left behind.
So there isn't time for mechanical equilibration here, basically.
Yeah. So one can start the migration even after 1000 MCS or 2000 MCS to allow the system to equilibrate and then start the migration. So all of these changes can be done. For example, you in this code you can add uncomment this line and say if MCS is greater than, let's say, 500, then start migrating. So I'll close this and rerun this simulation again. Oh, seem to crash. Hmm. So it will wait until the step reaches 500 and then start migrating. So you see it's stalled right now. So yeah, now it starts to migrate. The blue
You might
cells.
have to increase the speed of the basal cells and the other cells.
Oh yeah. Those things can be changed
Yeah.
accordingly, and
Now there's a big disparity between
right.
at the back.
Yeah, of course. Let's see, changing both of them. So you saw that again. It starts migrating after 500 MCS. So now if we also change. Where was that part? So we can also, for some reason, this part of the code did not have the basal side, basal external potential also. So let's have both of these at -20 each and starting at 500 MCS.
Will your superficial cells get left behind in that case?
Could be. That's interesting to see. Let's see. Or are they pulled along? So is everyone able to play with the model, or has anyone tried? I think Aritra has tried.
It works for me.
Yeah. So you see, because we have imposed only the imposed that only the basal part of the cells will be imparted with the migration plugin external potential. So that's why you see the shearing effect of these cells, and that is kind of the reason why we set the external potential for all of these cells to be quite low, so that uh, these effects would not happen and the cells would take their time to equilibrate and then migrate.
I guess if we could make the breaking
Large.
stress of the links higher.
Mm-hmm.
I tried -5 instead of -20.
Mm.
I'm trying to see if it's going to break or not. It looks almost breaking, but not quite.
Okay. 5 and 5 and FG and the FPP links, we can increase their maximum distance. Wnt and FGF lat, we can increase that distance to say 200. And sorry.
No, go ahead.
And so was everyone able to follow? We changed this focal point plasticity plugin parameters, Wnt and FGF lat. I'm just changing the maximum distance parameter from 40 to 200, so that the links have a longer range and they don't break up, hopefully.
Abhishek,
And sorry.
did you
Yeah.
have that video of the activated notch for them to see later on?
Yes, yes,
Yeah,
yes.
maybe we can.
I'll go. Yeah, I'll go to that after this. Um, so let me try opening it in the player, and I also changed the um, lambda vecx, the migrated potential vector in the x direction to -5 for both Wnt and the basal cells.
These observations are actually kind of interesting and useful because we're actually studying in the lab now more of the changes that take place in the neuromass that are preparing to deposit, and between what it, so we're also learning, beginning to change this model so that we can incorporate deposition into it. But what's interesting is from the biology, we are learning that as cells prepare to deposit, the adhesion molecules that they express also seem to change. And there's some that may come on that may not only affect adhesivity, but other properties of the cells that determine how effectively they migrate. So there's a very interesting thing, transition that's happening as cells prepare to deposit. The other thing that's not actually captured in this model, and I think that we might want to go back to, is in the actual system, there's an oscillatory behavior where the leading end stretches more and then the back catches up. So to what degree this is a natural outcome of the mechanical coupling versus waves of activity that are influencing the contractility and behavior. So these are all things that are sort of under active investigation in the lab. And then as we learn more, we'll go back and incorporate these things in the model.
So there was this interesting thing that started happening, where you see that the skin layer, it broke off, and the FEP links broke off, and the superficial cells are sort of filling that gap, and the Wnt cells are also filling that gap. So these are the unintended consequences that we try to mitigate in our model with the various parameters. So yeah, it's quite cool to see how the system evolves with different kinds of parameter values. Yeah. So now after increasing the FPP maximum distance, you see that this link did not break between the Wnt and the FGF lat cells, but and they are migrating. They are stretched out a bit, but they are migrating together. But then there is something else happening here, and because of which these cells are their shapes are getting weird now. So yeah, I'll stop this simulation here, and I'll show you another interesting um, example that we were able to recapitulate. So this is kind of like a question. And this is an actual, we try to recapitulate an actual phenomenon that someone else found out in a different lab. And I won't explain what it is yet, but if someone could tell me like what's going on here and what parameters can be changed to achieve this two rosettes fusing together. I'll play it again. So these two rosettes fuse together while
Abhishek, you don't
migrating.
have the actual video of
The
the,
the
that would have
actual
been nice to
video,
show them.
no. Yeah, maybe having that might would have been actually mm, nice. So does anyone have any idea like what simple parameter to change to achieve this?
So what Abhishek is showing is what happens actually in an embryo in a situation where notch signaling is exaggerated in the entire primordium. And just as background, it was interesting because we had always thought all these years about notch signaling simply being involved in this process of lateral inhibition, which, as I explained earlier, helps to restrict ATO1 to a central cell. But experiments like this have actually reveal to us that it has this other complementary role in determining the cohesiveness of the cells and various parameters that affect cohesion. And
Yeah.
so you
So
can
yeah, so basically notch, what it does is the other group had shown that overexpressing notch increases the adhesivity between these cells, and so that tends to make huge rosettes and fewer rosettes. So both of those aspects can be captured here by just increasing the relative adhesivity between the lateral domains and the apical domains. These are the only two parameters that we changed. You can try playing with various ranges of values and see whether you can recapitulate this effect. Yeah, and we already kind of did this, not the lambda, the link strength, but we changed the maximum distance parameters. So you can also try changing the FEP link strength or the lambda, that is the strength with which the links contract, and reach a certain or want to reach a certain target distance. And you can also increase or decrease the target distance parameters so that you can check what effects do they have, each of these have on the rosette formation capabilities. So can someone like put it into the chat what will happen if we what will happen to the number of rosettes or the size of rosettes if we increase the lambda or the link strength?
So by increasing length strength, you mean increasing the contractility or increasing the breaking strength?
So increasing, so if we keep the target distance or the maximum distance parameters consistent, and then increasing the link strength itself would lead to more breakages in the neighboring links, and also that would allow for smaller rosettes but more number of rosettes. Yeah, so that's uh, what we get. And similarly, if we increase the or decrease the target distance parameters, so then again you have smaller rosettes. So these are things that people can play with in that model. And please add that external potential on the basal layer like I showed. I can show that again. So that was something that was missing. So please copy paste this module into the next line and just replace Wnt self.Wnt with self.FGF-bas.
Okay, maybe slow down. Let's do this together.
Right.
So I'm supposed to go into my code and copy lines 53 through 57?
50 to 57, yeah.
Okay.
50 to 57.
Yeah.
And then copy and paste that in the next couple of lines.
Mm-hmm.
And just change where it says for cell in self.cell list by type on line 59. Just change it to self.FGF bas, the basal cell layer. And so there are other commented out modules here which you can try out. Here what we did was in line 68, I'll just uncomment this out. So here what we did was between steps 5,000 and 10,000, we imposed the stalling phenomenon of just the wind cells. So we had the cell migration, wind cell migration drastically reduced, and that led to and also we had reduced the cell fluctuation amplitude or the temperature to 10, and mostly the change in the migration speed allowed rosettes to coalesce together, as I showed in that video. So you can try these things out. And then by uncommenting the next part of the code from line 78 to 86, we again resumed migration after 10,000 MCS to the initial levels. So the initial level of migration speed or migration force for wind cells was -2. We reimpose that. And don't forget to save your changes. I'll just comment it out. Anybody have any questions?
That worked for me. Aritra said they got it to work. Anybody else get it to work? So you mentioned at the beginning this NetLogo model, and that you were implementing that one in CompuCell2. I'm always very interested in replicating models. And so I was saying it might be not a bad hackathon project to do that little NetLogo model as a CompuCell model. I was going to ask one thing. Of course, this is already a big simulation and it's fairly slow. So you wouldn't want to do this on your desktop machine, but you might do it on a cluster. If you were going to actually try to go into 3D, so you had the full simulation in both sagittal and lateral directions, what would the biggest challenges be in trying to combine this kind of sagittal model that you have here in CompuCell with the full lateral model that you have in NetLogo? Have you tried that or thought about that yet for what it would involve?
So I think we briefly tried it out. Apart from the computational cost, which if we run on a cluster, it should be okay. But the thing which was a bit challenging was how to assign these relative cell-cell interactions with each other, because the top view model, which Ajay briefly showed in his last slide, and this sagittal view model, these have different cell-cell interactions or the parameters in terms of the contact energy parameters and so on. So since there were a lot of cell types, that was particularly challenging to get just right so that we could have all of them. But I think with enough sort of iterations, that should be doable. I don't think it's an impossible challenge. I think it should be doable. Yeah.
Okay. Did you have any other more material you wanted to present today, or was that?
Yeah, it was just another.
Great. Why don't we
Yeah.
go ahead? Why don't we
Right.
talk about that? Go ahead, please.
Oh, okay. This was just about repeating the second exercise after like the model has had time to equilibrate, and then repeat changing the FPP link strengths or the target distance parameters, and do they change with respect to like whatever you observed in 2 exercise 2, where they start out with that modified parameter. And so the idea is that when cells are themselves or the model is trying to reach an equilibrium point, um, they will have imposing these constraints will have different effects versus when they have already reached an equilibrium. And so that is also kind of cool to know and understand whether our model simulation really requires that or not, or whether there are some unintended non-physiological effects that are happening. So people can try that out if they want. And yeah, that's it. That's it for me.
Great. Well, that was really, it's a very sophisticated model. A number of people have been interested in modeling migration on a deformable substrate. And that's something that you're doing here. So the methods that you're showing here are ones that the people who are interested in doing that might want to look at.
Yeah, I've shared the paper link in the chat. So anybody interested can have a look at it. And I have also put up the codes on GitHub, both the NetLogo model codes in that paper as well as the CompuCell model codes. The various variations, the different variations that we have put in that paper, all the codes are there. So anybody wanting to uh, modify it or recapitulate the results, they can do that.
So we
That's
have,
great.
yeah,
Go
so we
ahead.
have 3 directions that we'll probably continue to develop the model. One is to include the reaction diffusion stuff that helps to create the patterns of FGF activity so that we have both of those things influencing where and how these rosettes form. And then the other thing we only very briefly alluded to, that the notch signaling actually has a very interesting effect on regulating adhesivity between the cells and other properties that influence the rosette formation. So that's the other direction we want to go with these models.
Well, it's impressive. I really appreciate you taking the time to show it to us today. And for our students, some of the things you saw this morning, the links you'll see next week. So you can come back and revisit this seminar after you've seen how to build links, although they're not that complicated to do. And so I want to thank you, Ajay and Abhishek, for presenting this today.